and then the last 15 in genetic epidemiology. And this was because I was fortunate enough, as I was doing my neurology fellowship, to become affiliated with the Framingham Heart Study. And on October 25th was there as about 500 participants came back for the 75th anniversary of the Framingham Heart Study. About 6 years ago, I was invited to go down to Texas. The state of Texas is about as big as the country of France, but at that time didn't have an Alzheimer's disease research center that's NIA affiliated. The NIA has a big program for the last 30 years. And so I was invited to start up an Alzheimer clinical as well as clinical research, as well as population and basic science research program there. And this has been a lot of fun over the last 6 years. And about 3 years ago, we got recognized as a ADRC, partnering with center that's right on the border between the US and Mexico, and so deals with a lot of issues of Mexican-American and immigrant health. The Mexican-American Hispanic population, we use the word Hispanic, but that's a very diverse group. And the Mexican-American Hispanic is the fastest growing and the fastest aging group in the US. Right now, about 2 out of 5 Texans are Mexican-American, and actually Texas as a whole is, if we take non-Hispanic whites as the former majority, it is already a minority majority state. That means everybody who's not non-Hispanic white is over 50%. But today I will not be specifically talking about what we do or have at the Glenn Biggs Institute or at Framingham. Rather, Framingham was also an entry into collaborations like the Alzheimer Cohorts Consortium, and one that is really a consortium of the willing over the last 15 years and has enabled me to form ties and partnerships with people like Monique and Irfan. And this is what we call the CHARGE Consortium, Cohorts for Heart and Aging Research in Genomic Epidemiology. So I'll be touching upon some collaborative work across CHARGE, where I have been part of the neurology working group for these 15 years. Instead, today, I was sort of giving a more topic-focused overview of should we think about dementia in the oldest told. A lot of our current approaches to dementia are using the human model of dominantly inherited Alzheimer's disease or CADASIL to kind of extrapolate and say that if there is certain pathways implicated in familial dominantly inherited Alzheimer's, probably it's also relevant in older onset Alzheimer's. And increasingly, we are finding that this is not true. And particularly if you're talking about dementia in the oldest old, why is that important to study? What do we know about the biology of dementia in the oldest old? And can this teach us about risk and resilience in everybody with dementia? And by focusing on this group, what do we understand about life course risk and resilience factors? And then, the ultimate goal is promotion of brain health. So, needless to say, everybody in this group knows that the fastest growing group is the over 85. And given that the baby boomers are now entering that age, we expect starting 2030 that this is a very fast growing. In addition to this, this is also the group where dementia risk is highest. And so by 2030, 1 in 2 people with dementia will be over the age of 85. A lot of our research, including epidemiological studies, undersample this group. And so we can define this a little variably. We don't know that there's a hard biological line at age 80 or 85 or 90. Some people may be oldest old at 80, and some may be young at 95 or 100. We have people in Framingham who drive down from New Hampshire, something like a 2-hour drive, and say, no, you don't come and see us. I like the change. So, and already you've heard about the fact that dementia risk is going down. This is Claudia Sadizabal presenting in 2014. This also means that the average age at which symptoms appear has gone up from 80 to 85 in the Framingham data. And so why is this important? Also, as I said, we know that 5-year incidence keeps increasing, and data from studies like the 90+ study headed by Claudia Cavas and Maria Corrado has shown that we wondered whether it levels off. For certain diseases like Parkinson's, most, or say even myocardial infarction, if you survive to be that old, you probably have good lifestyle, good genes, and you don't seem to keep increasing in risk. But in dementia, it looks like the risk continues to increase even beyond age. In terms of societal costs, public health, and economics, this is the group with the least informal supports, because often the spouse may have died. Often this is a woman who has survived, and the highest cost per patient because there is multimorbidity, right? People not only have dementia, but they also need to remember to take their heart failure pills and their anticoagulants for atrial fibrillation and so on. So what do we know about the biology of dementia in the oldest old? Is it just more of exactly the same thing, or is it different in some key ways? We know dementia is just a syndrome, and it just means that some acquired cognitive abilities or behavioral or emotional control abilities have declined, and they affect the person's ability to do what they need to do. We began recognizing that dementia is not just due to one thing, that there can be multiple causes. But something that is often not as much appreciated by people who are not epidemiologists or clinicians is that, of course, having Alzheimer's doesn't protect you from having vascular dementia. Turns out it doesn't protect you from having dementia with Lewy bodies either. And so, the medicine has that principle of Occam's razor that multiple symptoms try to, but then you have the Hickham's dictum that a man or a person can have as many diseases as they damn well please. And so we recognize that particularly in the oldest old, often there is not just one etiology. In fact, almost everybody, I would say over 90%, have plaques, tangles, amyloid, vascular injury. And the more you recognize the diversity of vascular brain injury, the higher the proportion of people who have some signs of it. And about 30% each have alpha-synuclein in a distribution that qualifies as the Parkinson's disease to dementia with Lewy body continuum. And about 30% have the TDP43 deposition, this entity that used to be called hippocampal sclerosis and is now recognized as late. So those 4 big things, as well as some things that we understand less well, like primary age-related tauopathy and argylophilic grain disease, pathologically these are there. We are still just beginning to define what is it that we see in life that corresponds to what we see in pathology. And we know that in between all this is the lifelong resilience reserve, maybe starting before life in utero, right? And then you have all these other environmental factors, things like inflammation, things like COVID. In the US, you have programs like Recover, C4R, that is about 14 cohorts looking at before and after the pandemic. As well as some efforts we are involved in using 7 Tesla imaging and other cohorts. But the point I want to make is that this is just one example. There are Rush and many other pathological series, including CFAS, that have shown that if you look at each individual, it's an exception. There are only 2 people in the first 140 who had only a single pathology. almost everybody has more than one pathology, and therefore multiple etiology dementia is in fact the most common type of dementia. And we're just beginning to have biomarkers for the various types of dementia. So, of course, in starting 2018, there was the famous ATN criteria that requires the presence of amyloid and says it can be preclinical, prodromal, or clinical if amyloid and tau are present, along with neurodegeneration. And more recently, you have the draft criteria where comments are still being invited that finally recognizes that there is a vascular component, that there is an inflammatory component and an alpha-synuclein component. And so I just touch upon where we might be with biomarkers, because we don't have universally agreed upon biomarkers for these undeniably important contributors to the clinical dementia. So of course, amyloid is something that has driven the field, but we have to recognize that if you look, of course, there is a much higher risk if you're amyloid positive of developing clinical Alzheimer's. However, even in this data, over a 5-year period, over 90% of people who are amyloid positive at the beginning remain free of clinical dementia. And if you sort of look at people, particularly in the oldest old, about 40% of people over 95 with normal cognition are amyloid positive. And we can say, yes, if they live long enough, maybe they will get Alzheimer's. But at 95, you're not looking at a life expectancy of 20 years. So we do need to think in the prevention of dementia beyond the age and amyloid boxes. And we also need to understand, are these pathologies just additive, sort of incidental, or synergistic? And which pathologies are the most common? So dementia with Lewy bodies, the clinicians know its presentation is slightly different. There are earlier Parkinsonism features, there's more executive visuospatial changes. REM behavior disorder may occur several decades earlier. There can be early autonomic dysfunction, dizziness, falls, orthostatic blood pressure changes. Very famously, anxiety and mood changes came to the public attention because Robin Williams suffered that and may have led to his suicide. And it's very hard to treat because if you treat the Parkinsonism, the behavior worsens and so on. There are biomarkers on FDG PET, on the cingulate sign, on DAT scans, and then there are other biomarkers being thought of. One is this seed amplification, but also things like looking at autonomic nerves in the heart, skin biopsy, and the REM studies. Late is more recently recognized, the star DNA binding protein 43, interesting protein also implicated in ALS FTD, seems to be related to stress. Some interesting data from the cardiovascular health study pre-publication, where over nearly 2,000 people, TDP-43 levels were higher in African Americans, and we are looking at whether this could be related to higher societal and racism-associated stress that they see. But basically what you see is the hippocampal atrophy that may be asymmetric. You see these TDP-43 granules, the stress granules. Normally this substance is in the nucleus, but it goes into the cytoplasm and forms these. Clinically, these are older patients, slower decline, but we rarely make the diagnosis in an antemortem way. We typically say, oh, this is a variant of Alzheimer's, and now we are trying to see if we can distinguish based on imaging or other things. There do seem to be some genes. Interestingly, many of these genes are genes like progranulin and ApoE that actually change the age at onset of Alzheimer's. So these may just be kind of resilience-related pathway. We know that having co-pathologies can affect the clinical picture of some of these. And of course, vascular, that everybody in this group knows. And the definition of vascular remains evolving. And if you take the broad definition that you just need to have cognitive problems, disease affecting the vasculature of the brain, and evidence of damage to the brain, And today we take evidence of damage mainly based on MRI. But as these criteria, like the STRIVE-2 criteria that were recently published from the University of Edinburgh and other collaborators, as these expand, as you start looking at things like cerebrovascular reactivity and microinfarcts, you find that a higher and higher proportion of people have vascular injury. So that the right question may be how much vascular injury, what type of vascular injury, rather than if somebody has vascular cognitive impairment or vascular dementia. So the increasing recognition is, let's call it vascular contributions to cognitive impairment and dementia. It's not VCCID because that is sort of awkward. But even in early onset Alzheimer's, you have a substantial proportion of vascular. But particularly if you look at the oldest old, this blue line is around age 85, you find that the proportion of cerebrovascular disease, this is a slide from Pete Nelson, is kind of greater and may account for a fair bit of the increasing risk that we see. The other thing is that some of the traditional risk factors for dementia start having, maybe you could call it paradoxical, but they start having associations that are unexpected when we're talking about the oldest old. This is a review by Deborah Gustafson in Lancet that shows data that all the cohorts have shown that in midlife, obesity is bad for you. But then pre-dementia, you actually have, starting about a decade earlier, a drop in BMI. And not everybody necessarily, but if you look at the group as a whole. And so when we do a dementia risk score, having a low BMI, less than 19, actually increases your risk of dementia in the group as a whole. Emma McGrath, who's now at Galway, looked at Framingham data and said, can we see what is the biggest risk factor among the traditional vascular risk factors at different ages for dementia? And remember, the average age of dementia onset is 85. And you find that at younger ages, it's systolic blood pressure. Almost throughout life, it was diabetes. But in older ages, it's actually what I would say markers of this lifetime exposure to these vascular risk factors. So at older ages, it's stroke. It's rather than the systolic blood pressure per se. And so here, arterial stiffness is thought to occur even before the onset of hypertension. And in over 7,000 people from Framingham, if you divide it by quartiles, arterial stiffness was a very good predictor of dementia. And this has been estimated that hypertension control could actually significantly decrease population burden. But if you look at blood pressure from midlife to late life, again done by EMA, you find for sustained high systolic blood pressure increases late life dementia risk. But a steep decline in blood pressure from mid to late life in normotensives also increases dementia risk by about 250%, 2.4 hazard ratio. So, and these don't act in isolation, of course. Mostly people these things happen together. That's why you have things like the stroke risk profile, cardiovascular risk profile. And sometimes in somebody with diabetes, for instance, we now well recognize that it's not so much blood sugar control that's important as blood pressure control. So if you're looking at risk of something in an epidemiological way in persons with diabetes, perhaps what you need to look at is not just the hemoglobin A1C, but what their blood pressure has been. And similarly, if you look at, say, obesity and hypertension, this is very old data, you find that waist-hip ratio and high blood pressure together have a synergistic adverse impact on cognitive measures. So this has become, from a time when it was thought that you can't have dementia unless you have plaques and tangles in the brain, now everybody, whether it's the Alzheimer's Association or anybody else, says it's Alzheimer's disease and related disorders. And another mantra has become what's good for the heart is good for the brain. And to some extent, this is true in terms of reducing risk of stroke. You know, these are life's essential 8 as described by the American Heart Association, the usual things, but also adding sleep. And Monique showed dietary inflammation is a marker of brain aging, and she showed that with the Grim age here. Deborah Mello and Lent showed this with MRI measures. Matt, some years ago, had shown this with not just sugar sweetened, but even artificially sweetened beverages. The MIND diet in observation studies seems to be associated with lower risk of dementia. Some recently, just at the last AAIC, I know all of you probably heard that the RUSH study, which is where Martha Claire Morris first did initial work with the MIND diet and dementia, did a randomized controlled trial, and the MIND diet didn't make a difference, right? That's always disheartening. But one reason may be that doing it over 5 years isn't really going to give us an answer, because perhaps it needs to be MIND diet starting in midlife that's going to have a benefit. And this is Claudia looking at DHA. We have some Framingham data on about 2,000 people with ceramides, again showing that C22 was associated with lower white matter hyperintensity, but I'm not mentioning that here. Physical activity, again, yes, physical activity is good for the heart and the brain, but perhaps not exactly in the same way. They seem to be, for the brain, it seems like any amount of physical activity is good. So you don't have to meet the heart guidelines of 30 minutes of moderate to vigorous a few times a week. And recently, data from Eric showed that even not being sedentary for 5 minutes in every hour, getting up and walking, may be good for cognition and the MRI. But again, these things occur together, and this is from about 200,000 people, not work that I've been involved in, but showing similar to what we have been doing, that if you take clusters that include both risk factors as well as markers of disease like MI, you find that clusters are better predictors, and the more morbidity you have, the higher the risk of developing dementia. Interestingly, this was the doubling of the dementia risk was actually greater in people who had a lower genetic risk defined by the polygenic risk score for dementia, now about 90 loci. And this was done not in the oldest old. Most of the average age of this group was in the 60s when risk factor was determined. So clearly there's more work to be done in this area. Another paper, again, from the amazing UK Biobank, about 500,000 people, and frailty being defined, it can be defined in different ways. Here being defined as a composite of weight loss, tiredness, physical activity, gait speed, and grip strength. What you do see is that not only is frailty associated with a higher risk, but as the number of individual frailty criteria increase, this risk is increasing. But what does this mean? Is this causal? Is this just a biomarker of some things happening in the brain as they are happening in the body? So how can we sort this out? One thing would be if we had a nice biomarker, like we have amyloid for certain things, if we had it, or even say BNP for heart failure, if we had a nice biomarker of each of these morbidities or pathologies, or say cystatin for the renal function, right? If we can put them all together, perhaps we can say in this 90-year-old, these are the things that are wrong. But of course, it's not that easy to sort out biomarkers because multiple systems have impact in one biomarker. If you take something like neurofilament light that comes from neurons, your kidney function still makes a difference to how much neurofilament light is circulating in your blood. But here are some approaches now NIH is clearly recognizing that we need to use precision medicine approach and searching for biomarkers. The two obvious ones are amyloid and tau. We're still working out, I know Harp has been looking at different tau variants. And so the flavor of the moment is perhaps p-tau 217 that was used in the donanemab studies and correlates very well with amyloid levels. In the donanemab study, it was used to correlate with amyloid status, not with tau status, and predicts also cognitive decline. Less clear are ideal biomarkers for inflammation synaptic and for the other many genes that we are discovering, as well as for the other diseases that I just described. And what are good surrogate markers, if not biomarkers, for environmental factors, for behavioral factors, like how well have I slept? Am I jet lagged? Is there a biomarker that can tell? So this is a recent, things talking about amyloid tau, but also things like GFAP, which is an astrocytic marker, YKL40, which is a microglial inflammation marker. But the question again becomes, for amyloid tau, there are about 5 to 6 assays that are competing. Probably one will come out on top, and what systemic factors affect that as well. In Framingham, for instance, we just took all the old data that was available on a bunch of biomarkers, and then, yes, many of them are individually related, but what if we put them all together and use different machine learning methods? And we found about 41 candidates, but 10 that came up with all the different methods, usual suspects like A-beta 4042, but also some things like IGF-BP2, leptin. So interesting to follow up. And of course, if you take an even larger canvas and say here, let's take the SomaScan. This is an older version with only about 1,200 proteins. And this is in the charge cohort. It's not just Framingham. This is across multiple cohorts. Discovery in 8,000, replication in about 9,000, O-link validation, related proteomic platform, not exactly congruent, in about 3,000 people. We found certain consistent patterns. The orange is the replication. The blue, the blue is replicated using Bonferroni, the orange using FDR. You can see most of these do have good replication and certain interesting markers, inflammation like CRP, but also Nectin-2 are turning up and interesting to follow. Imaging patterns of atrophy are another thing that are emerging for aging, for differential diagnosis. This can be X amount Alzheimer, X amount dementia with Lewy body, X amount aging. And something that's being recognized is that while we think of cognitive reserve, and we may think of cognitive reserve as something that needs to be determined very early with education and other things, the number of synapses, your early childhood nutrition, there could be cognitive resilience that may be something that we can alter throughout our life. And so in this data from the National Alzheimer Coordinating Center pathology data on over 4,000 brains, 17 pathological lesions were aggregated to say this is the damage, and then you estimate what the cognitive function would be based on these pathologies. And people who have better cognitive function than expected are considered to be cognitively resilient. People were defined as having reserve based on things like your gender and on education status. And so they would life means you have high reserve. But then your slope of decline can still be different based on perhaps the degree of resilience. Now, this is just a concept. We still need to understand that this resilience, what is the biology underlying that? And some of the studies that are trying to understand the famous NUN study. Maggie Flanagan moved in February to the San Antonio, and we have all the NUN study brains and materials, and the nuns would very much like to invite collaboration from all over the world. We are trying to digitize all this data with some philanthropy, both the digital pathology as well as the digital antemortem data, which is amazing data, and I can talk during the coffee break. And then the nuns, like Framingham, are mostly white. And so we need to have diversity. The San Antonio Heart Study began in 1979, and this year we are bringing them back. They've had intermittent exams in between, but this will be focused on the brain with MRI, PET, and other measures. And this is led by Claudia Setizabal. What are the things that might determine resilience? Something that, again, we don't know biology necessarily. We have some clues, but in terms of association, Joel Salinas showed that at the same brain volume, cognition was better in people who said they had someone to talk to. This was among 2,300 persons in the Framingham study, and that perceived loneliness. You know, when we're talking about social support, there is the strength of your network. There is also whether you perceive loneliness or not. And the perceived lonely had a higher risk of dementia. So the Minister of Loneliness in UK is ahead of the curve. Other countries need to do that. So using genetic loci, we are able to do polygenic risk scores, which are now beginning to show a separation and are also predictive of transition from MCI to dementia, the prognosis part that Dr. Hoffman was referring to. We are finding genes here, PLC gamma 2, that might be protective for all of these different pathologies, like ApoE2 may be protective for multiple pathologies, 4 may be bad for multiple pathologies, and may also be increasing longevity. So in this decade of healthy aging, it's important to recognize that this is data from Framingham that Gina Peloso shared. Regardless of whether you had high genetic risk, changing lifestyle factors seem to reduce your risk if you had good lifestyle factors by the same 50%. Of course, the starting point was different. So polygenic risk, but another way of looking at this, of course, every risk gene, the opposite allele can be taken as a resilience gene. But what if we try to define genes that may be important for resilience and less so for risk? And so Stephen Glatt and others, we have some ongoing work. And what we show here is that if you define people based on their Alzheimer genetic risk score, who, however, are cognitively normal, we assume they have some resilience genes, right? And so if you compare these high risk resilient controls to people with high genetic risk who did develop Alzheimer's, you can find resilience genes that are different from AD risk genes. You can also do a polytranscriptomic risk score based on transcriptome data. So these are interesting and worth following. Coming back to the CHARGE consortium that I defined and its sister, the cross-cohorts collaboration, which looks at non-genetic associations, very grateful to all these studies. About 6 years, 5-6 years ago, we began this collaboration and have looked at things like lung function as a marker of accelerated brain aging, or non-alcoholic liver disease, particularly liver fibrosis, various other ongoing papers that are submitted or in press or resubmission, including at different things like the heart, inflammation, renal function. The big advantage of having multiple cohorts is that the results become more consistent over time. And now we are adding more diverse cohorts, such as the San Antonio Family Heart Study and MESA and Strong Heart, which is a Native American cohort. The big thing I think that we will need collaboration for is when we are trying to understand the biological pathways underlying these associations that we are confirming. And this is very nice example where when you looked at the metabolites, Vincent and Cornelia Van Dyne, you found, of course, the expected thing that fish intake increases DHA, but less expected is, say, physical activity or the lack of physical activity results in changes in certain metabolites, that the same metabolites are also associated with poorer cognitive function. Again, looking at these biomarkers, in this case, amyloid and tau, on PET imaging in Framingham, we found that women and men of the same age behaved a little differently if we were considering postmenopausal women. When we looked at premenopausal women, age was the strongest determinant of how much tau you had in your brain. But when we looked at postmenopausal women, women at the same age had higher burdens of tau. What exactly this means? What is the mechanism? Is this replicated in other cohorts? These are things that we can look at. Same way, when we looked at, again, the PET data, when you look overall at non-alcoholic fatty liver, no association. But when you look at concurrent liver fibrosis markers, the FIB4 index, then consistent association with amyloid and tau. And to find these kind of, you realize the numbers have gone down from about 170 to about 40. So I think this is true, but we need large consortia to find such small effects. So to summarize, half of all dementia will soon be in the oldest old. Multiple etiology dementia is the norm. There are a number of risk and resilience factors, and they may present as multimorbidity and need to be sorted out. And so large collaborations are needed to identify these nuances and understanding the biology, which can lead to more targeted and more effective prevention trials. And this is the large gathering at the last CHARGE meeting that was held in San Antonio just about 3 weeks ago. And I hope to see many of you all at the next one. Thanks.
That's great. Thanks very much, Sudha. I mean, you make the case very strongly that this is a multi-system, multi-pathology, disorder, and certainly what's seen in clinical practice without access to PET scans and everything else. You've got a whole bunch of things in there. So that makes it very difficult. I mean, even the polygenic risk score is polygenic risk score against all these multiple different pathologies and biology. So how do we unpick all that?
Very good point. I think what we do is at an individual patient level, maybe we don't need to unpick all of it, but by unpicking parts of it in studies, we are getting new insights into biology, right? Like now there are drugs targeting SP1 and PLC gamma. The polygenic risk scores, it's a very good point that we call them Alzheimer's scores. And when I began, our genetic colleagues would be like, we need to have, and this is true, we did this, I think we made a mistake. The ADGC initial thing was, let's take people who have escaped Alzheimer. And so let's take only the oldest people as controls, because we don't know if a 70-year-old is going to develop Alzheimer's. As epidemiologists, I was saying if one 70-year-old does not have dementia, another 70-year-old has dementia, the two are different. We can compare them. But that wasn't the route we took. And so what we, I think if we have a polygenic risk score, this is really a polygenic risk score for dementia, and it's really a polygenic risk score for dementia at age 80. We don't really know if we take oldest old, maybe there's a different polygenic risk score, but we have polygenic risk scores for blood pressure. Again, maybe blood pressure at 55 is determined by a different set of genes than blood pressure at 85. And I think some of that work is being done now that we have these huge consortia for things like blood pressure and BMI. So Mendelian randomization may be one way, but I do think we need to have phenomic biomarkers as well, whether these are plasma biomarkers, whether these are, imaging or arterial stiffness or something. But which will be the most valuable? If we are, as clinicians, we may not care as much about those that won't change our treatment. So we may just pick biomarkers that we can treat. And we already do that, right? We chase blood pressure or cholesterol more aggressively than we chase domains of cognitive function, because our treatment isn't going to change based on that.
Yeah. So finding a biomarker that is truly going to be predictive, it's predictive, but predictive in a way that's going to lead to a change of treatment. Exactly. That is a very big hurdle.
So MARC-VCID, some of these efforts are trying to say, let's find biomarkers that can be surrogate markers for intervention studies.
Yeah, so that's a very high bar. Yeah. Questions from the floor? We'll start with that.
Very nice presentation, sir. The concept of resilience, I think, was interesting. Do you think this is a active mechanism, or is it just lack of pathology or slow aging that we observe?
Okay. I think there is mechanisms promoting resilience, because some of these, like SP1, PLC gamma, they do seem to be, so on the one hand, as you said, there can be anything that's a risk factor. Its absence is a resilience. But that's why it makes it complicated. But one idea of this study design was to try and use the polygenic risk score to define it. And we didn't find ApoE is the strongest resilience gene. We did find other new loci. Ultimately, if we do mouse studies, maybe we'll understand the biology.
Sudha, it's so exciting to have you here, and it's such an awesome amount of work that you've been part of and led. I wanted to go back to one of the real challenges is to understand when we need to start our search for the origins of, or the seeds of, dementia in later life. And what's your view, A, about how we should conduct this? I mean, looking for disease decades before it happens is a hard thing to do. And B, what you think we've learned from those efforts that have been made to find early changes that may be associated with the seeds of the pathology of later life.
Certainly, it's a big problem. I think we also recognize that even at a biological level, even at a genetic level, development can give us clues about response to neurodegeneration or plasticity, right? We know that Notch3 mutations are associated with just white matter hyperintensity distribution, that progranulin, has, or say TREM2 has Nasu-Hakola and has Alzheimer's. And so there's with things like white matter hyperintensity, for instance, studying this across, there are student cohorts, and you're finding that some of the genes associated with older life are also seen at younger ages. Can we have, I think we do need, as somebody pointed out, and there's an increasing recognition of collecting data around pregnancy, around early life, regularly in all these cohorts, even if they are enrolled at 25. Transgenerational is good. As far as even trials go, it's not impossible for us to follow up. Before Framingham, people didn't treat hypertension. it took 20 years between 1948 for the first papers to come out showing that smoking and high cholesterol and high blood pressure are bad. If we accept that not every study is going to give us answers in 5 years, certainly where prevention is concerned, we have established cohorts that can be followed up, but we could do interventions and then expect to have answers by 2050. It's better to have an answer by 2050 than not to have an answer at all.
I mean, you've touched on something that's really pertinent. Again, we talked about these cohorts, maintaining the cohorts over. I mean, funders work on a 5-year cycle, maximum 7 years, and you have to say this is a 30-year, 40-year project.
Right.
And it's sort of we're really struggling with that right now in the UK, because which cohorts do you support long term to get the answers to the questions? You start early and you have to wait and you have to watch.
Right.
Even if you say following up people in trials, you have to do long term follow up. And of course, once the randomization has been done, there's no longer randomized. It becomes observation.
Right. And some level, I think all of us need to be willing to say, and I will, if it means giving up Framingham to maintain UK Biobank, that's okay with me. But at least some of them have to keep going on.
Yeah. We're going to get you as a referee on all our grant proposals. Okay, thanks very much. Brilliant.